Hello everyone, welcome back to the channel. Today we're going to be asking a very simple question. Were endogenous retroviruses created? Do they represent designed units of DNA function? If you've consumed any amount of creationist versus evolution content on the internet, you have heard the term endogenous retrovirus. I'm not going to go into great detail in what they are here. I've done a whole video looking at the parasites in your genome. I'll link that in the description below. What I want to do in this video instead is address a very specific creationist model of endogenous retroviruses. There is one creationist who has just taken this mantle and has become the endogenous retrovirus guy. He is standing for endogenous retroviruses. And of course, the person we're talking about is Donny Budinski at Standing for Truth. He's written a book on the topic. He's done multiple debates on the topic. And for reasons largely unknown to me, he's put out 2 back-to-back videos on endogenous retroviruses within just the past couple of weeks. I don't know why this topic kind of like fades and we start talking about genetic entropy or out of Africa or other things, and then endogenous retroviruses inevitably comes back. But for whatever reason, it's back and he's making content on it. And one of the reasons I kind of wanted to make a video addressing his model specifically is that I've noticed a lot of other creationists have kind of adopted it. Like it's spilled out, it's now bled out into the rest of the creationist community, and they are now using his arguments. And so what I want to do is take his model. We're going to steelman it. I'm going to give you exactly how he presents his model, and then we're going to address what I think are some of the fundamental flaws of it. Again, these 2 videos aren't anything new. These are things he's been talking about for a long time. But what really kind of prompted me to do this video was this last little clip that he did in his most recent one, the dismantling the critics, in which he takes this kind of like comical approach. So I'm going to play you the clip, and then we're going to get started.
Be a biblical creationist, because even your most militant of critics like Dr. Dan Stern Cardinal here, who lives and breathes fighting young earth creation, he doesn't have good responses. He has no response for how these functions can come about. Yeah, they can say co-option all day. They can reiterate their story over and over again, but that doesn't make it science. These guys are bloated with evolutionary philosophy. It's evolutionary philosophy for breakfast, for lunch, for dinner, for dessert, and as a bedtime snack. The evolutionary philosophy is leaking out of their ears and nose. They've been preaching evolutionary philosophy for so long that they can't even see that they're preaching this evolutionary philosophy. So I am going to reshare, but I just want to make sure that the audio is shared. Okay, here we go, guys.
Have we directly observed an ERV that integrates and then acquires a beneficial function in the host? The answer is no. Have we directly observed an ERV that integrates and then acquires a beneficial function in the host? The answer is no.
The answer is no.
That's all!
Okay, so remember that clip. It might come back. We're going to start off here with Donny's model. We're going to call this the endogenous retrovirus created model, the ERV created model, and it basically has 3 points. The first is that all endogenous retroviruses that are fixed within the population are created units of DNA that have function. Okay. Importantly, what fixed means is that every single individual, everyone in the population has this endogenous retrovirus. Okay. Everybody's got it. That's what we mean by fixed within the population. If that's the case, then he is stating that that endogenous retrovirus was created in place and it must have some kind of function. Okay. Second is that ERVs only resemble viruses. They're not actually viruses. They just look like viruses because they act in immune response. He also notes that like 80% of endogenous retroviruses are actually just solo LTRs. And I think it's important to note here that solo LTRs don't act in immune response. That's not what they're doing. Solo LTRs serve exclusively as promoters and enhancers because that's what they do in the viral genome, right? That's what is in that long terminal repeat, that LTR region. It just serves as an enhancer or a promoter. So he misleads a little bit here with this point because one could ask, well, if solo LTRs are only serving as promoters and enhancers, why do they need to look like a virus promoter or enhancer? The genome already, the host has its own promoters and enhancers. So it makes no sense to me that God would need to design promoters and enhancers that just happen to look like virus promoters. That's irrelevant. That's not even the point here. I just want to point that out. Regardless, they only resemble viruses because he thinks that they all act in some way in immune response. And then lastly, and this is what we're really going to focus on, is that polymorphic ERVs are actual ERVs. He accepts that endogenization of viruses can happen. He accepts this because it has been demonstratively proven, right? Like we have seen viruses integrate into genomes in many species, and we have polymorphic endogenous retroviruses in our own genome. And again, what we mean by polymorphic is that some individuals in the population have it, others don't have it. So he accepts that when they're polymorphic, as opposed to being fixed, they are actual viruses that have endogenized. So he does accept that endogenization can occur. So that's what we're going to focus on. We're really going to focus on this polymorphic ERVs being actual endogenous retroviruses. Okay, so this is the structure of the critique. First is we're going to talk through model inadequacy, that his model cannot distinguish fixed and polymorphic sites. Second, we're going to talk about insertional age, the age at which that polymorphic endogenous retrovirus actually entered the genome. We have some that are really, really old, and I'm going to talk about how we know that. And then lastly, the big kicker, the thing that he thinks that no evolutionist can provide him, is that there are polymorphic ERVs that have acquired a function. Now remember, polymorphic ERVs he accepts are actually endogenous retroviruses. And that whole little clip, that cute little clip he just played, was claiming that we don't have any evidence of this happening. It doesn't happen. It's just storytelling. Somebody didn't read the literature. Somebody didn't actually take the time, the time you took to put together that cute little clip. You could have been reading the literature and you would have known that we actually have polymorphic endogenous retroviruses that have acquired function. And I'm going to show them to you. And then we're going to revisit your little clip. So, do variants fix? Are they capable of going to fixation? This is a question that Donnie needs to ask himself. If we look across the genome at the different frequencies at which these endogenous retroviruses occur, I've plotted a couple of them here from two different studies. So here, this is the actual genomic coordinate along this column. These are the various different endogenous retroviruses. This is what flanks them. And then these are three different measures of their frequency. So from three different data sets, how frequent do they occur? So we know that they're polymorphic. So notice that some of them, really low frequency, 0.003. Some of them have really high frequency of 0.95. So 95% of individuals have these ERVs. And so what we see here is that there's a range of frequencies, some really low, some really high. Over here, this is an even larger data set using the 1000 Genomes Project, and the different colors represent the element's frequency. So the really light, so down here, these are the really light colors, very, very low frequencies. As you get closer and closer to 1, you get really, really dark colors. Okay, so Donnie, the question is then, what about these endogenous retroviruses that exist at 0.99% frequency? Like they're 99% of individuals have them. Is it possible that at some point in the future, it will be fixed? Everybody in the population will have it? That means at one point you accepted it was polymorphic, which means you accepted it's an endogenous retrovirus. It's actually a virus that endogenized. But is it not conceivable that in the near future, given that some of these are greater than 95% represented in the population, it will become fixed? At that point, then, could your model tell which one was a virus versus which one was created? How would it distinguish those two things? Is it capable of distinguishing those two things? For this model to make any sense at all, you would have to claim that these endogenous retroviruses that you accept right now are ERVs could never go to 100% frequency, could never fix in the population. Because as soon as one of them fixes, your model is invalid. You can no longer distinguish between those two things. That's something to think about, because surely we can at least both agree that alleles can go to fixation. Okay, next is that we have really, really ancient polymorphic endogenous retroviruses, some that were inserted greater than 1 million years ago. Let's talk through how we know that. So when a virus first inserts into any cell, any one of the genomes of any one of your cells, the 5' and 3' LTR regions are initially identical to each other. So I'm showing here the 5 and the 3'. The black is just the viral, the actual protein coding sequences of the virus. Doesn't matter what they are. All we need to know is that those two LTR regions are identical. They're repeat regions, and they act as promoters and enhancers for that actual gene. Okay, so once that gets inserted into your genome, let's say it actually makes its way into the germline to become an endogenous retrovirus, over time, as it gets passed on from generation to generation, those two regions start to accumulate differences, right? They start getting a little bit different from one another. And the rate at which they acquire differences is exactly equal to the rate at which mutations occur every single generation in individuals. They match the pedigree mutation rate. And the reason for this is that it's a virus. It's not doing anything for the host, right? And so there's no pressure to fix those mutations when they occur. And so what we can do is directly estimate the age of insertion given the number of differences that have accumulated between the initially identical 5' and 3' LTR regions. When you do that, when you do that method, you find that some of the endogenous retroviruses in humans, especially those that are at really high frequency right now, have been in the population for up to 1.8 million years. And this, again, is using a pedigree estimated mutation rate. So if Donnie accepts that polymorphic endogenous retroviruses do actually represent viruses that have inserted, then he needs to explain how it is that these viruses appear to have been in the population for almost 2 million years. Because again, we have some that are there that are really, really old. Now, he can reject the mutation rate, but he would need to give evidence of the pedigree estimated mutation rate being wrong. We have a very, very recent study that just verified the same exact mutation rate that this 2016 paper used to estimate this 0.6 to 1.8 million years. So every study that's been done has come to a very similar rate, right? There's not a gigantic range in estimated rates. They're all around 1 to 2, 10 to the negative 8, right? Like that's the range that we get. So he would need to somehow present an argument that the rates of mutation in those regions are much, much faster than what we expect. Now, there are some studies that shown that gene conversion can happen in the LTR regions, right? So you can't just naively calculate the rate. But what this 2016 study did, and what many studies since have done, is they have accounted for gene conversion, right? So this is a thing you can account for in your model because you know what it looks like when genes undergo gene conversion, and you can avoid those regions that have high recombination rates, okay? So it's not some intractable thing that we can't account for the fact that gene conversion occurs. Lastly, and this is the kicker, is that we know of polymorphic ERVs that have functions. Again, to remind you, Donnie accepts that if ERVs are polymorphic, then they really are endogenous viruses. Okay, so then demonstrating that any polymorphic ERV could have a function is then showing him endogenized retrovirus can acquire a function. That's the thing he's been asking for this whole time. Here it is. I want to read an excerpt paper from 1993. This particular gene variant that we're going to talk about was first discovered in 1975. We've known about this thing for a really long time, but in the 1980s, we actually characterized it as an endogenous retrovirus. So I want to read this excerpt. So they write, natural resistance to subgroups of retroviruses has been found as a polymorphic trait in several experimental species. In mice, one of the strongest genetically determined resistance traits was first identified in Japan. This is the 1975 paper, and designated FV4 because it was the fourth friend virus resistance trait characterized. The same resistance gene was discovered independently in wild mice in California. FV4 resistance is inherited as a simple Mendelian dominant trait mapped to mouse chromosome 12. After breeding of the FV4 resistance onto laboratory mouse strains, it was shown that inheritance of FV4 is associated with expression of a cell surface antigen cross-reactive with ectotropic murine leukemia virus, envelope glycoprotein GP70. FV4 resistance also co-segregated with a replication defective ectotropic virus-related proviral sequence, presumably the gene that encodes the envelope-related cell surface antigen. What does that mean? What is that saying? So up here is the little diagram. So the MULV, this is the leukemia virus that they're talking about, the friend virus. And you can see it's got the LTR, GAG, POL, and ENV, the envelope protein gene. That's what it looks like. Now, this particular gene variant that they're referring to, this FV4, is a broken endogenous retrovirus. It doesn't have all the components. All it has is a partial pole, a full ENV, and only the 3' LTR. It's missing everything else, okay? Not everybody has it. Importantly, it is polymorphic, and it is broken. But what it does, what they just described that it does, is that envelope gene will get transcribed. And if it gets transcribed, it will create just a dead envelope. There's no virus in it, nothing in it, just an envelope, and that envelope will bind to the cell surface proteins. And so what leukemia is trying to do is also bind to those proteins so it can inject itself into the cell. This empty envelope blocks that receptor. It takes up the receptors so leukemia can't enter. And therefore, it acts as an immune response, despite the fact it is very clearly a broken endogenous retrovirus, and it's polymorphic. Not everyone in the population has it. Importantly, it acquired the exact kind of function you would expect an endogenous retrovirus to acquire. And this is something, again, that he seems to be surprised when these endogenous retroviruses have functions related to immune response. Right? Because one of the things that actual viruses do is suppress immune responses, right? That's what they do. They also bind to cell surface proteins. That's what this one is doing. They also have LTR regions that act as promoters and enhancers. That's what the solo LTRs do, right? They're not doing anything that's surprising for an endogenous retrovirus. So here we go. This is exactly what Donnie was asking for, a polymorphic endogenous retrovirus, i.e. one that he accepts is an actual endogenous retrovirus that conveys resistance to a leukemia virus. So, in summary, Donnie proposes that fixed endogenous retroviruses represent created DNA units and should all be functional. He accepts that polymorphic ERVs are actual viral insertions. We have shown that many of these variants are at high frequency, very soon to fix, right? At 0.99% frequency, and at any moment could fix in the human population. His model would then go from calling them retroviruses to calling them created units of DNA function, right? In a generation. So any model that would just flip-flop like that is a useless model. We have shown that these viruses have inserted a very long time ago. These polymorphic endogenous retroviruses have been segregating in the human population for almost 2 million years, using the originally identical LTR sequences on either end as calibration points to calculate how long ago they diverged. And then lastly, and this is really the main thing, this is really the main crux of Donnie's argument, is we have shown that endogenous retroviruses that are polymorphic, that Donnie accepts are endogenous retroviruses, have secondarily acquired a function. Something that Donnie has claimed cannot happen, has never been shown, no one has ever presented it. And quite frankly, this study that I cited here is old. It's been around since 1975. That paper that I showed that was actually characterizing the function has been around since 1993. All right. We have known about this for a very long time. What I also didn't do is go through the fact that most of the human-specific endogenous retroviruses from the HML2 group, the group that has the most complete retroviruses, and ones that he has pointed to as being functional, are very likely also polymorphic. Because again, they're very young, and we know many of those are polymorphic. So it's very likely we will find that some of them have functions as well. And so with that, let's watch that clip one more time, shall we?
He doesn't have good responses. He has no response for how these functions can come about. Yeah, they can say co-option all day. They can reiterate their story over and over again. No! But that doesn't make it science. These guys are bloated with evolutionary philosophy. It's evolutionary philosophy for breakfast, for lunch, for dinner, for dessert, and as a bedtime snack. The evolutionary philosophy is leaking out of their ears and nose. I am going to reshare. But I just want to make sure that the audio is shared. Okay, guys, I think that's the perfect place to end it.
My recommendation is to be sure you really go through the literature. And I don't mean that as a slight. This is something I tell my students. This is something I was told by my mentors, is to be sure you really thoroughly go through the literature before you make a claim. Because somebody at some point is going to check you on it. Thanks for being here. I'll catch you guys next time.